Okay, so you'll need to be in Schoology. We're going to talk about what we're starting. We're starting the animals. This is the last major topic we're going to talk about this year in biology. Okay, what we'll be doing is we're going to talk about the animals. You have your STAR test. Then we're going to talk about anything that we really struggled with the most, as far as like body compositions, body build, all that stuff. Then we're going to be doing dissections. All right. So all that will start afterwards. So this is our last major lecture stuff, all that. So if you'll get inside the very bottom folder, it's a purple folder. And when you look at it, it's going to say Animal Diversity and Evolution. That's the very bottom folder in the main Schoology. And inside that folder, you'll see this stuff. Again, the top one is the PowerPoint. You need to go ahead and click it to download that in a minute. Don't do it right now, because we're going to talk about what else you see in here. So the PowerPoint is here. The note packet, that is what I gave you when you walked in. It's just like the notes we did earlier in the year. But the change is going to be, as we talk about stuff and we do the lecture, you can try to take notes and answer some questions, but it's really going to be a lot faster than you can do that. So what you're going to do is you're going to have part of class time to be able to answer the questions. Okay, and I'll tell you whenever we're done, kind of what you need to do, how to answer them, and that kind of stuff. But you'll use the PowerPoint. It's just like we've done in the past. We're just not going to take a day where we go back and use them to review. We're just going to answer the questions as our own little review. That make sense? If you lose that packet, you will need to print it, because once it's all complete, we're going to turn it in for a grade. Now the rest of the stuff, just like we did with the plants and the fungi, because everybody's going every which way in the spring, you've got all the academics, you've got FFA stuff, you've got UIL stuff, you've got games and sports and all this different stuff you're going to. I do every folder by date. Okay, so when you look in here, just like the plants were, but instead of a folder with PowerPoints or videos in it, we're going to have lectures. So we're going to have lecture on the 20th, 21st, 24th, and 26th. Okay? The 24th, we'll also have a little short video to watch afterwards, because it's really a very important topic. Most of the time, you'll have a question over chordates on the STAR if you have a question over animals. So we're going to watch a little short video to kind of reinforce what we talk about in class. The 25th, you have a STAR test. Okay, so what's going to be is there's going to be a video that you can watch on your own time when you get home. That way we're not doing stuff, you're not worried about missing biology for STAR, because that's not fair to you. So we will have nothing in class because you're going to have the STAR test that day. But then that evening or the next morning before school, sometime you're going to have a video you need to watch. Does that make sense? So it kind of gives you a little time after the STAR test to not have to immediately say, oh, I got to go do homework. That video will be on there. Now, the last one is a primate evolution. Okay, and that is a lecture. That lecture will be posted in there as well. So if you're going back and you're answering the questions, you need a little help from the lecture, go watch the lecture. All right? If you find it on the PowerPoint without the lecture, do it that way. It's your choice. Everybody clear how it's going to work? That way if you miss a day, you know, hey, I missed the 24th. You come here, you look in that folder, you click that, and it will have in the 24th what we did. Also, it's always on Google Classroom telling you what we did that day. All right. So go ahead and download the PowerPoint. Then you can follow along if you wish. You don't have to while we're doing a lecture today. But we're going to talk about the first section, and then you're going to have a few minutes in class that you can work on those questions. All right, so you don't have to do it all outside of class. So first of all, when we talk about animals, there's lots of things that we use to classify animals. In other words, we want to be able to talk about them. How are they built? Why are they this way? How do they compare to this animal? Are they more advanced, less advanced, and so on? So the two main things that we use are going to be things that have to do with their body plans. In other words, exactly how is their body structured, how does it develop, and so on. And the other thing is going to be the differences among those phyla that are created by body plans, is what we're going to talk about. Does that make sense a little bit? The phyla basically are the divisions. When we talked about plants, remember we had conifers and angiosperms. Those were phyla as well. So phyla is just a classification or division of our animals. So the first thing we're going to talk about are all the things that have to do with an animal body plan. What do we look at with animals? Because we just talked about plants. We looked at roots and leaves and stems a little. Talked about their berries, how they reproduce, right? Pollen, all that stuff. Well, animals, we're going to look at certain things too. And the first thing is we're going to look at their levels of organization. And what's meant by that is when you talk about levels of organization, are they multi-tissue? Do they have lots of different things they're built out of? And I use humans, for example, because hopefully most of us know we're human. Right, Brock? I don't know if I am anymore. I think my brothers are a gorilla. But I think I've got human tendencies to me. So we have skin tissues. We have other cells that help protect us, right? Immune system. We have blood. We have brain cells. We have other nerve cells. Muscle cells. We have all these different levels that our bodies put into. So we're a multi-level organism. The lowest animals are going to have less levels. The more advanced animals, which humans are considered to be the highest advanced animal on the planet, doesn't mean we're the smartest or bestest, but it means we have the most levels and the most diversity among us. Okay? So levels of organization. The next one is body symmetry. Body symmetry, has anybody ever said, hey, that's symmetrical? Or have you ever been told, hey, that looks symmetrical? Yeah, what are they trying to say? What are they telling you when they say it's symmetrical? Maybe the same size, same shape, right? So if you look at it, it kind of looks the same, it looks symmetrical, right? Well, it's kind of the same idea when we look at animals. Except what we're doing is we're considering if we put a cut or a slice through them, it's called a plane. So if we look at an animal, and no matter where we put that plane, each side or each part of that animal looks totally different from the other part. Okay, that's going to be one type of symmetry we look at. If we look at an animal and we put a plane through it, and then we put a plane in another direction, and another direction, and another direction, and that animal always looks the same on each side, no matter how we cut it, that's going to be another type of symmetry we'll look at in a second. And then the third one is if we put a plane right down the middle, each side looks the same. But any other way we slice it, it doesn't. Okay? So let's talk about those symmetries in a minute and kind of look at how they work. The next thing is going to be definition of germ layers. How many of you think germs are just small bacteria and stuff? Okay, so what else is germ used for?
Medicine.
Do what?
Medicines.
Medicines?
Or is that science?
No, I mean medicines, we'll use the term germ, but it's talking about kills bacteria or small things. Generally in our life, okay, if you're just a normal person, whether you're a student in high school, elementary, or you work in the workforce somewhere, germ is used as a very small, tiny cell that can infect or make you sick. That's what we call germs, right? But in science, germ layer is referring to a tiny cell. That's what a germ is. It's just a small cell. So when we say we're going to talk about germ layers, we're going to talk about how many different kinds of cells make up layers of that body. Does that make sense? So it's not just tiny bacteria when we say germs. You got to understand, scientifically, germs referring to small cells and then layers of tissues that it makes. Then we'll talk about body cavities. What is a body cavity? What do you think that is? Sean, you have an idea? What would they think with body cavity? What would you think they're talking about? Okay, compare it to a cavity in a tooth. So what's a cavity in a tooth?
Like the decaying area?
It is a decaying area. So if part of a tooth has decayed, Yanni, what happened to that decayed part? If part of the tooth broke down and decayed, where did that part go? Is it gone? You don't know?
Dead.
If that part's dead, so what does it probably do? That part's gone, right? So what does that leave?
The germ.
A hole. And that's what a cavity actually is talking about. If you have a cavity in a tooth, they're saying you had decay or broken down tooth matter that died and fell out and left a hole in your tooth. That hole lets bacteria get in there and do it more, and it eventually gets to a nerve and hurts, right? So they fill it. That's why it's called a filling. They fill the hole that's in your tooth. Well, when we talk about body cavities of animals, it's kind of the same thing, except it's not dead material that's missing. It's just a space in that creature. Do they have a space in them? Do we have spaces? Yeah. What do we have in our spaces? We have a huge space inside, and then inside that we have our organs. We have our intestines and our lungs and our heart. All that's in a cavity. Our brain's inside a cavity. It's a space inside your body. So when we talk about that, that's a big deal on how to organize different animals. Next part, embryological development. What do those giant words say?
How
Embryo. How babies develop. Yeah, that's good. That's everyday terms, right? I mean, you don't walk over there and say, hey, does your puppy have good embryological development? You don't say those words. You say, hey, did your puppy develop right? Is it okay? So embryological development, we need to store that in our brain as how did the baby grow? That's our everyday terms, isn't it? It's embryological development is how did the baby grow? And we're going to look at how that baby develops. What does it do when it develops? How long does it develop? At what point does it start to grow stuff? And so on. The last three kind of go together and kind of don't. Segmentation. Have you ever seen an earthworm?
Yes.
What do they have all down them? Little lines, right? What do those little lines do?
Separate.
They separate. Those are all little sections of that worm. There are little spaces in there. It's segmented. Okay? In the upper animals, segmentation involves forming a head. My head is a segment of my body. My chest is called the thorax. It's a segment. It's just a part of my body, right? So segmentation is, are they divided up or are they all kind of the same creature? You know what a sponge is? Sponges are animals. They're filter feeders. That means they live in the water. Water goes through the holes, comes out the center hole. They trap the stuff behind, absorb them, get their energy. They don't go hunt. They can't move. All that good stuff. But they're still an animal. But they're unsegmented. If you look at one part of the sponge, it's basically the same as the other, right? It's not divided up any. It's just kind of a sponge. Fake sponges are made to resemble real sponges. So if you look at a fake sponge in your house, it really looks kind of like a real sponge anyway. One part looks kind of like the same as the rest. The next part is cephalization. If segmentation is occurring, like in an earthworm, but a true kind of head area develops. With the central nervous system, possibly eyes and a mouth, it's all kind of in one location. That's called cephalization. Okay? And then the last one, of course, is limb formation. How is it built? How does it grow? And how many? Y'all know what the term bipedal is? To let you know, this is an anatomical term, but pedal refers to your foot. Have you ever known that? No, you never had a bicycle? Yeah? What are those things called that you put your foot on? Pedals. But they're really pedals. That's where you put your foot. Because a pedal refers to your foot. So what is bipedal? What is about 2 feet? So what do you think? What do you think? What creatures might be bipedal? Us. Yeah, humans are. Things that walk on 2 feet, right? A quadruped. What do you think those guys are? They walk on 4 feet, not 4 legs, because we kind of have 4 legs, right? Because that can get pretty touchy. Is this a leg? Is it an arm? What about a bat, right? So legs is kind of a touchy word. So when you say quadruped, you want to say they have 4 legs, basically. Or 4 feet is what you want to say the best. Because when you say legs, it can get touchy. So ped refers to foot. So quadruped actually means they walk on 4 feet. Which normally in us means what? You got 4 legs. Okay? So quadruped. Are there animals that are quadrupeds? What's a good example of a quadruped? A dog, what? A bear. Now, bears are pretty cool, aren't they? They walk on 4 feet, but what can they also do pretty well? Walk on 2. They don't walk as well, but they can walk on, but they can stand up, and right? So there's different animals that can do different things, and it's all due to their limb formation. How's that limb built? All that good stuff. All right. So let's talk about this symmetry. Those little glass-looking planes that are in there are basically what I was talking about, the way they cut them. So if we put this plane, we cut it this way, then this is one side, there's the other, they essentially look the same. So we cut it this way. Now the left and right essentially look the same, don't they? No matter how you cut that creature's going to look the same on each side. That's called radial symmetry. What does radial refer to? Come on, Math Whiz. The radius. And what is radius? It's referring to a circle, isn't it? So radial means a circle. So that thing basically looks the same in a circle. No matter how you cut it, it's going to have a left and a right that looks the same. Okay? That crayfish, you cut this guy down the middle, you're going to have a left and a right that looks the same. What if we cut it like this? Is it going to look the same? No. That side look totally different. What if I cut it kind of at an angle across here? They going to look the same? No. So that guy is called bilateral. What does bi mean? Two. What is lateral? If I tell you to move laterally, what does that move? Sideways. Yeah, so it means side. So it's two-sided. Radial basically means multi-sides, doesn't it? It looks the same in a circle. This guy looks the same on just two sides. And this is going to come with some other terms called ventral. That refers, whoops, hit the wrong side. Ventral, that refers to the mouth, okay? Now that's the one that gets kind of catchy with animals. Remember we did the sharks? Y'all remember that, say, asking if the mouth was up front or along the underside? Ventral, you remember that stuff? It was saying, where is the mouth at? This comes into play with the crayfish. The ventral surface is where its mouth is, which is what we would call the belly, or the underside, isn't it? What about us? Where's our ventral surface?
At the bottom.
Right here. It's right here, where our mouth is. So it's on the front. And that's really what anterior means. So ventral is going to be a little different depending on the creature and how the body's built. But anterior, posterior, that's front and back. Dorsal is the back to us, and ventral to us is the same. So anterior and posterior are the same as ventral and dorsal on a biped, on us. But on a bear, he's going to have an anterior and a posterior, and he's going to have a ventral, which is his belly, okay, and a dorsal, which is his back. So the best way to think of those two is exactly that. Dorsal means back. Ventral means belly. So if I look at a dog, and I say, where's the ventral surface of that dog, Sarah? You don't have to go, okay, his mouth is kind of here, but it's on it. Find his belly, that's the ventral surface. Okay? What about a fish? Fish are kind of weird, aren't they? So where's their ventral? You don't find their mouth, because it could be on the front, could be underneath like a suckerfish. What do you find?
Belly.
Their belly. That's their ventral surface. Okay, so that kind of takes care of some of the problems. All right. Now, symmetry-wise, you got to think about a sponge, because there's not many things that are called, they're called unilateral. It means they only look the same when you look at them. If I cut a sponge, in reality, that sponge is going to look different, because it's going to have different size holes, right? Don't sponges have different size holes all in them? Plus, they're going to be a little bit different shape, because, and we'll see a picture in a minute, but a sponge may have 2, 3, 4, 5 pieces to it. So if you cut it in different ways, you're going to end up with different parts, so it's not going to look the same. That is called unilateral or asymmetrical. Asymmetrical is usually the common word they use. So that just means it has no symmetry. All right? Now, we're going to talk about these layers, germ layers. It's talking about the layers of those tiny cells made. We got to talk about, and by this point, y'all probably heard this. Has anybody ever joked with you and said, hey, your epidermis is showing? Anybody ever done that to you? By this age, usually you've had a granddad or a grandma, somebody thought they were cool. They go, hey, Brock, be careful, man, your epidermis is showing. Usually put it on little kids. They get all nervous. They think they didn't zip their pants or they did something wrong. They go, oh. Does anybody, what's your epidermis? Anybody know?
Your belly button?
No. And that's usually why they pull that joke on you, right? It's your skin. Epidermis just refers to your skin. It's a scientific term for your skin. Dermis is a layer of tissue. Epi means surface, so it's your surface tissue. Epidermis. Okay? But usually by this point, somebody's pulled that on somebody in my class. Hey, your epidermis is showing, and we go, oh my God, what I gotta do? You know? And then you're like, oh, it's just your skin, and they like laugh, and they get their joke in. My granddad tried to pull it on me all the time, so I just had to laugh and I had to act like I didn't know what it was every time. Okay? Make his day whenever he got to make you laugh. So, when we're talking about derm, like epiderm is the outer layer, we're going to talk about the layers of these things as they develop. Again, you're not going to remember every one of these words. You're not going to do that on the test. I don't expect you to know all this. There's a class in college called zoology. They study just animals for a whole semester. Okay? So these words are not going to be expected to be memorized and learned, but we want to be familiar with kind of, hey, they're talking about derm. I remember that was like layers or something. Okay? We want it to kind of come back to us a little. So these terms, ectoderm, ecto means outside. So as it develops, the layer of tissue that develops on the outside is called the ectoderm. That make sense? The next one is meso. Meso refers to middle. So as the creature develops, the tissue that layers in the middle is called mesoderm. It's a dermal layer, it's tissue that's developing in the middle of something. The last one, endoderm. What do y'all think? What does endo mean?
Front.
Okay, so let's use our hints.
Inside.
Ecto was outside, meso was middle. Ecto or endo probably means inside, doesn't it? And it kind of sounds inside. Endo, inside. So endoderm is the innermost layer that as the thing develops. So these pictures are just going to kind of show you, and there's going to be some names. Again, I don't expect you to remember these words, but I would really hope that you recognize them. And there's a big difference. On the test, when you recognize it, you'll possibly remember what it talked about. You just couldn't define it, probably, but you need to go recognize it. So, whenever this creature basically has a true ectoderm and an endoderm, and then this mesoderm is just kind of like body filling. Those are called acoelomates. They don't have a true body cavity or anything, it's just kind of tissue in there. A means there's not really one. Coelom is that's the body cavity. So this guy doesn't really have a true body cavity. Alright? This guy is called a pseudocoelomate. Remember, a coelom is a body cavity. Does anybody remember pseudo? We've talked about it a couple times this year. What does pseudo mean? We talked about it with some amoeba stuff. There's pseudopods. Your white blood cells have pseudopods. What is a pod?
Something that holds.
Well, we talked about pads and pods are the same. It's your foot. But amoeba don't have feet. They have pseudopods. Pseudo, the word means false or fake. So this one, does it have a body cavity? Yes, but it's kind of a false or fake body cavity. These guys develop basically a circular space in between the outer layer and inner layer of their tissues. So it's just a circular space. Roundworms have these, like the stuff that comes out of your dog eat. Real smooth roundworms, not earthworms. And the next classification or things they would look at would be coelomates. That means they have a true coelom. So if you can look at this picture, see that space? What does that space have around it that this space doesn't? Anybody tell me? Look real close at the space, the coelom, the cavity. The one on the right's got something that makes it different from the left, from the middle one.
It's segmented.
Well, that's segmented on the outside, but look at the body cavity, right there where the kind of pinkish tissue is.
Yeah.
That space. What is different from the one on the right and the middle one?
Has a protecting like layer
Where?
in between like that yellow tube and the.
Yeah. So see, if we look at this one, it's called a pseudocoelom. See, there's the mesoderm. But then there's the endoderm and the space, the cavity meets up, doesn't it? But if you look at this one, this cavity is surrounded by mesoderm, isn't it? There's a space and mesoderm's all the way around. It's got like a protective layer between it and the endoderm. Okay? Those are upper level creatures, like us and all the other animals, okay, and birds and earthworms, segmented guys. So again, that's an advancement. That's considered an important thing because it's, what if something happens in one cavity? Does it automatically get in all the other ones? No, because it's got a protective layer, right? If something happens in here, it's going to spread everywhere. That one doesn't even have a body cavity, so these are advanced. All right. These words we're going to talk about, we're not going to look at the stuff on the right. What we've got to remember is deuterostome and protostome. We want to hopefully remember stome. You remember stomata on the plants? What were stomata? Remember the word, but what were they? They're stoma. What are those? No, it's on a leaf. Does that help y'all any? They're on the underside of leaves. Yeah, they're basically the little mouths that let gases in and out of a leaf. So a stoma is basically referring to a mouth. This is our stoma. So when these words have stoma on them, we need to kind of think, hey, they're talking about a mouth or an opening. That's what we really need to remember with these. Now, if you were in zoology, and we're studying this, a deuterostome is called a certain thing because if the anus develops from the blastopore, that means this opening that forms, when you first get fertilized, okay? So there's a sperm and an egg in any animal on the planet that gets fertilized, makes a zygote. That zygote immediately starts going through mitosis, dividing, right? Cell division. And it divides to 40 cells. That 40 cell ball is called the blastula. From there, all of our tissues start developing and growing. All of those 40 cells do a different job. Some of them become maybe your nerve tissue, some your blood tissue, some your nerve cord, some your bones, all that stuff, okay? So we look at those ectoderm, mesoderm, and endoderms to see how do these guys develop? How do they grow? And if they grow where that opening makes an anus, then it's called a deuterostome. If the opening makes the mouth, it's called a protostome. Now, are you going to remember all that? No. It'd be awesome if you could just hear it and talk about it and remember it. But we need to recognize when we see the word stome, hey, it's referring to the mouth. And then maybe you can remember a little bit about where it develops and where it comes from. Okay? But that's just the way that we use to classify all the animals, is how did their mouth and anus develop? Where did it come from? What tissue layer developed into that? Okay, segmentation. Remember, this is not symmetry. You get confused with symmetry a lot. Symmetry is how is it built in a radius, bilateral, remember? We talked about how you could cut it if it looks the same or not. Segmentation is not that way. This picture that's on here leads you to believe that. Don't. Okay? Segmentation is saying, is that body divided into sections? Like us. We have appendages, legs and arms. We have a torso. We have a head, right? A grasshopper hopper, he's got a head, a chest area, and a tail, doesn't he? We call abdomens. So they're divided into sections. So these two guys on the left are segmented. If you looked at a fish, could you find a head? Sure. Could you find like its body? Could you find I can look at a fish and go, yeah, that's the body of the fish. This is the tail, that's the head. It's segmented. It's definitely developed into different parts, hadn't it? What about an earthworm? You can't find the different parts, but you can see the divisions on it, can't you? All right. Has anybody ever seen like a fluke or a leech or anything on TV? When you look at them, do they look all segmented? Or do they look kind of like one thing? One thing. They're not segmented. They're bilateral. Their symmetry is two-sided, but they're unsegmented. That means they just are one living creature. But they don't have like a head and a body and a tail, that kind of stuff. Right? That's what segmentation refers to. Cephalization kind of goes with segmentation, because if you're segmented, you're going to have body sections. But if you have a section that is developed into a head region, eyes, a mouth, whether it's a true brain, but kind of a brain-like organ, if all of those are in a centralized location, we call it cephalization. You're making a head. More advanced creatures are going to have heads. Even a grasshopper has a head, right? Does an earthworm? Not really. They kind of have a mouth and a lip. They have a little brain type area, but its segment doesn't look like a head, so it's not considered cephalized. But cephalization means growing a head. Okay? What about a snake? They're kind of built like an earthworm, aren't they? They're long and slender. Does a snake have a head? Yeah, they got eyes, mouth, everything's centralized in that spot, isn't it? So they are cephalized. Cephalization is having a head. Okay? Limb formation. Again, what do the limbs do? How do they form? How are they built? If you look at a whale's pectoral fin is built just like our arm, in the fact that it has a humerus, a radius, an ulna, and all the same finger bones, same number. Does it look like our arm?
No.
No, fins look different because of the tissues, and the bones are a little different lengths and things, but it's got the same number of bones and the same structure. So you can look at that limb formation, and you can see how we might be related way down the line, or we might have some common stuff. That's what we're looking at. Okay? If we look at bats, bats have wings, but the things that hold all the fabric or their tissues are fingers. Because they're a mammal. They have hair. They have all that stuff we're going to talk about. Hair, we call fur. They produce milk. Bats are mammals. So they have hands. It's just part of their lower limbs. So that's why we use the quadruped, right? Because that's really like an arm for a bat. But they walk on them a little. So we still say they're quadruped, even though they're more like an arm instead of a leg. So limb formation is important to kind of help us. All right. The next two things you're fixing to be using to answer some questions in the time we have left. The first thing up at the top, there's kind of a blue highlighted picture of it. Don't open it yet. The one on the far left, that is a sponge. Again, it kind of looks like someone's hand sticking up there. Not all sponges look the same, but they are all developed this same way. They do not have segmentation. They do not have body symmetry. They're asymmetrical. Their germ layers, they don't have layers to their tissues. They just have tissue. They just have cells growing together. They do have specialized cells. Some trap food. Some help move water through them. But they don't have layers and tissues and all that stuff. So they're considered a lower animal. As you move to the right, these animals are considered a little higher than the next. Okay? And we're going to come back, so don't go flipping real fast. This is a continuation of that same table. Moving annelids, mollusks, echinoderms, and then chordates are considered the highest living animals on the planet. Okay, so let's go back to this other page and kind of see how you read this. So the sponges, it tells us levels of organization, body symmetry, germ layers, tells us what they have. Obviously, if they have less than the one biome, they're probably less advanced. So if we look at the cnidarians, they have specialized cells, but they also have tissues. Do you know what a cnidarian might be by that picture?
Jellyfish.
Jellyfishes. Yeah, the jellyfish. Hydras, sea anemones, those guys are all cnidarians. They have radial symmetry. That means no matter how you cut them, they're going to look the same on right and left side. They have two germ layers. Okay, they're going to have an ecto and an endoderm, no mesoderm, none in the middle. They're called acoelomates. That means they don't have a body cavity. Remember we saw it's just all tissue. There's no real space, so there's no body cavity. They're not segmented or cephalized. They don't have a head. So which of these do you think are the higher order developments, evolution things? Levels of organization or cephalization? Cephalization. So segmented and cephalization are two of the most important things to be considered higher level. Mainly because we've learned they're going to have a head with a brain and a more advanced nervous tissues. They're gonna have coeloms, they're gonna have segments like a body, a chest, head. That's the segmentation part. Okay, so if we look at these guys, arthropods, those are your grasshopper hoppers, okay, and crayfishes, those type of guys. They're gonna be in the bilateral symmetry. That means if you cut them down one way, they look the same on either side. They have 3 germ layers. They have a true coelom. That means they have a true space in there that's surrounded. All right? But they're protostome. That means their mouth developed from a different spot, right? From that, not from the pore. But the main thing is they are cephalized and segmented. Now notice roundworms, they're basically the same except they're pseudocoelomates. So how are these, and they're missing segmentation, how are they considered more advanced than those guys? Well, that's where we look at how did they develop when they grew, what do their body tissues have, how do they get food or get nutrients, and do they have a nervous system type stuff. So that comes into play. And that's why they're a little less here, but they're still going to be a little more developed. And then flatworms, these are going to be guys like liver flukes. They're going to be your that gimmick. I just told you all a while ago. I'm getting old. Leeches. Okay. By the way, they still use leeches in medical fields. Not all the time, but if they reattach a finger or a limb, they'll put leeches on there to try to help draw blood to that area, because leeches suck blood or basically help pull it through, so it pulls it to the new area. Or if you have a real bad swelling from excess blood, sometimes they'll put them on there to get the blood out of that. They've used leeches for a long time in the medical field. Yeah, true story. Okay, so down at the bottom, main thing you're looking at, some of it's going to be cephalization. Do they have a true head area? If they have a head area, they may have a brain-like or a brain area as well. Again, annelids, those are segmented worms like earthworms and those guys. There are annelids that live in the ocean as well, so don't just think they're only earthworms. Mollusks, those are going to be snails, clams, those are going to be octopuses, squids. Okay, remember, you ever seen a chambered nautilus? Those are the cool dudes they show in the old, they like to say they're prehistoric, which they were around then, but they're kind of circular. Looks like a squid sticking out of a shell, but they swim around. It's a chambered nautilus, they're all in there. The next one are echinoderms. Echinoderms are going to be like your starfishes, okay? 2 things, flatworms and echinoderms have a real cool feature to them. Those creatures, if you cut them in half, guess what they do? They grow back the other half. So there was a problem for a long time. There was an area down, and I don't know if it's Louisiana or where, but I know it was in a swamp area. They were cutting leeches. They got them on them, they'd take them off, they'd cut them, throw them in the water. They just made 2 leeches. Because the bottom parts, the tail's going to grow a new leech, the head part's going to grow a new leech. They're going to have 2 leeches. Starfish do the same thing. There was an oyster farm around Japan. They used to pull it up, and of course starfish eat oysters. They drill holes in their shell and they suck the meat out of them. And so they were taking the starfish off there, cutting them in half, throwing them back in the water because they were still in their profits. They just kept getting more and more starfish. They finally figured out, wow, starfish can regenerate the other half. So they were just making more. That's a pretty cool deal. Wouldn't that be kind of cool? Have 2 of me, just cut myself in half, and tomorrow there's 2 contractions. You can get away with anything, man. But there are 2 creatures that can do that. And then, of course, when we get to the chordates, those are nerve cord creatures. That means they have a nerve cord, not just a spinal cord. Some creatures have a spine, some don't, but they have a nerve cord. Okay? So they don't all have vertebra columns. Those are the vertebrates. But these all have a spinal cord. And I say that because when we look at, this part that you're going to use, across the top are basically the same pictures we just looked at, except for the far area over here. This guy looks like a sponge, but he is called a tunicate. He lives in the ocean, looks like a sponge, but whenever you take him apart and dissect him, he's still an acoelomate. He's still not bilateral. He's radially symmetrical looking, but he's asymmetrical looking like a sponge, but he has a nerve cord. Sponges don't have nerve cords. So this guy is considered a chordate. And the rest of them you should know: frog, fish, turtle, bird, cow, or is that a deer? Yeah. Okay, so again, that tunicate, don't get confused and think, well, why is a sponge over there? It's not, it's a tunicate. But all the rest of those pictures should help you, because you're gonna need those to answer questions. There you go. That's where we're gonna start up tomorrow. You get working on those questions the rest of the class period. So you have about 10 minutes to get some done. Here's the deal. You may work with the people at your desk. Okay, so y'all can find stuff together, or maybe you can ask them if they know the question. You need to keep that out, because you're gonna need that PowerPoint. So you use that PowerPoint that I told you to have open to go answer your questions. And your partners by you. So again, I don't necessarily want you Googling it. I prefer you go find it on the PowerPoint. If you can't find it there, it's okay to go find the answer. But consult with the people around you, do a little peer tutoring, that kind of stuff going on, help you out if you need it. And those questions are gonna be just like the PowerPoint. The first questions are gonna be at the first of the PowerPoint, the second questions, and so on. So fill out that table, you're gonna need the first of the PowerPoint, not where we just stopped. Okay?